Hello, my name is Andreas Brammer. Thank you for coming across this video. Today I would like to discuss and share with you the method of self-raising we used in our tissue culture lab. So the topics of this video will include what exactly is self-raising, why it's used, when to use it, and the protocol affiliated with self-raising. So what exactly is self-raising? I'm willing to bet that when most of you hear the term self-raising, you probably gather this image in your mind. The image of some old guy outside freezing. In fact, this is actually more realistic to what self-freezing looks like once the process is finished. Cells grow and divide, and contamination is something that can occur quite easily, even if you're not trying to introduce contamination into the environment. And as a result, self-freezing is a technique that we use in order to preserve cells and to help keep them in long-term storage for further use. So while the video is playing, it's a good idea to give some background on why self-raising is necessary. As you can see in the video, the color of that tube is pink to red. Now, it'll change color to orange to yellow to blue. The reason for that is as cells grow and divide, they eat, they metabolize things. And the products of their metabolism cause media to acidify. Normally in tissue culture, we will see it be some intermediate shade of pink to orange and sometimes to yellow, but never will we see it usually go to blue as a color. So the very first thing we're going to do with this protocol is we're going to make sure our cells are confluent, because otherwise there's no reason either to split the cells or to freeze the cells. So let's operate under the assumption that the cells are confluent, say 90%. So in that case, we remove the old media, and we discarded that media into the liquid waste. we rinse with PBS in order to make sure that the T75 flask is rinsed and that any dead cells or cell debris gets collected, and we then remove it by putting that in liquid biohazard waste since it contains potentially dead cells and cell debris. We remove the PBS, and in its place we add 2 mils of trypsin. Then what we're going to do is we're going to place that into a COT incubator at 37 degrees Celsius for 5 minutes. Now, after this occurs, what trypsin does is it causes the cells to detach from the plate by cleaving the monolayer, the link in the monolayer protein that's present on the T75 flask surface. So to help encourage that process, once it's incubated for 5 minutes, we actually hit the T75 flask against a solid surface, such as a countertop. And as this process occurs, what you should notice is that the solution, if it's pink or red, you should notice it becoming more cloudy. And the next thing we're going to do is, if we were to keep this trypsin in the flask for too long, in fact, it would start killing cells because it would be cleaving proteins left and right. So we dilute the media by adding 8 mils of fresh media. We transfer it to a Falcon tube, and we centrifuge at 1800 RPMs for 10 minutes. So the reason that we centrifuge the cells is that when these cells have been centrifuged at 1800 RPMs for 10 minutes, what will happen is that at the bottom of the flask, albeit somewhat faint and not always easy to see, you should see the following. You should see a little white pellet, little white clear pellet at the tip of your 15 mil Falcon tube. And that is what we're going to be after and what we're going to be utilizing in the next and almost final part of this experiment. So I mentioned earlier that we were going to freeze cells, and I alluded to the fact that we couldn't do it using water, because if you think about it from common experience, if you put water in too cold of an environment, that water will freeze. Well, water is also present within the membranes of cells, so doing that would actually cause the cell to be killed. And this is where the hero of our story, DMSO, comes into play. So DMSO, or its full chemical name, dimethyl sulfoxide, actually is normally cytotoxic to cells. Normally it'll kill cells if they're in too long of contact with them. So we have to be very cautious when performing these next steps. So what are those steps? Well, those steps are going to be to remove the media from the falcon tube and leave a very, very, very small amount so that it's not disrupting the pellet. What we're then going to do is, in a separate Syra vial, we should have 100 microliters of DMSO and 900 microliters of fresh media added to it. We're now going to resuspend our cells in the remaining media we had from the falcon tube. So that mixture should be cloudy once you've done that. We're going to then transfer that mixture to the Syra vial, which contains the DMSO and the fresh media. And then what we're finally going to do is we're going to label the Syra vial with the name of our cells, that is to say the cell line, the date that this was done, and the name of the person who did it. So now for the actual last steps of this experiment. So this is called a Mr. Frosty, and if you were to take out the little metal piece here, you would see that there's this styrofoam, and there's actually a liquid in there. The liquid is isopropyl alcohol, or isopropanol. And if there's not any present, we would need to add it so that it covers a decent volume, but not all the way to the top. Then what we would need to do is we would need to put the metal piece back in. We would then make sure to put the Syra vials that we created with the cells into designated locations with the holes, and then place this in a -80°C refrigerator. So you might ask yourself, what exactly was the reason we did this to begin with? So the first reason is that if there is contamination that occurs at any point when doing cell culture, automatically that means that the integrity of the further experiments we do is null and void. So it's better to freeze cells periodically to avoid that risk and to keep fresh cells around. And the other reason is that if we were to just keep splitting cells, you would be doubling your workload each time. And at some level, that's just not feasible to deal with. Thank you very much for your time. I hope you enjoyed this video. Take care. God bless.